showcase the construct and experiment design menu system here. And the first thing I'd like to show you is the construct alignments. These are the alignments of all the structures in GPCRdb where the constructs are annotated. What you will be able to see is how the final construct looks, as well as all the inserts that have been added to the original wild type sequence. And in the schematic view, you'll also be able to see where there's been other forms of modifications, such as deletions and mutations. You'll also be able to see the fusion proteins that have been used, and that's the main features of the construct schematic. There's a similar wild type schematic. This is then taking the wild type perspective and then better show which areas have been deleted in the construct compared to the wild type sequence. For the experiments, the actual experiment values, such as which method has been used, how they've expressed it and purified and so on, that can be viewed in the actual experiment browser, which should load up here. This also has for all the annotated structures which chemicals have been used, the method, and so on, which can be of interest for the experimentalists who want to perform similar experiments. Right, then I would like to show you the design tool. Design tool works by inserting your target receptor. It then fetches data from both those of the same receptor structures that have been resolved from the same receptor, but also of all similar receptors in the class, to then give various forms of recommendations of what one might want to do. This, for example, is to select where to truncate the N-terminus. It's going to show you different points where one might want to truncate based on constructs that have been how the constructs look for the structures available. So there you can add these, and what you might appreciate is that you can add several sites, and this works for different concepts. For example, also the C-terminus. So here, and then it's going to make these combinations of, say, you now have two combinations possible that fulfill these cuts. Then one would also add a fusion protein and then add that along with the sequence, as this here. And you will want to choose where to insert this fusion protein. And here there are then suggestions based on what's been done in the receptor family or as the class as a whole. And you see the frequency and for which fusion proteins this has been used as a site. And so that's where I'm then choosing. And so here you might see there's two sites that are possible, and one can then add both of these. And what you'll see is that then the list of possible combinations has then increased. So that's for the step that we call the truncation fusion scan. So one would then get these constructs expressed and purified and test these and see which ones one might like and want to continue working on. Because the next step, that could be the mutant scan. And so this is more for making mutants that might be more thermostable. And so there's two lists here, and for this receptor, there are no known stabilizing mutations in our system. One might know themselves that they have one that they want to do. It's been proven by one's own lab, and you then add that. And do so. This is then in TM2 up here, but you might also want to look at our list of predicted mutations, which are there's various forms of predictions. Some are thermostabilizing, some remove glycosylation sites, and some are based on actual structures having had these mutations, so those are then added. These are, for example, the hotspots that they've been mutated. But there's also other forms of rules which you can read about in the paper. And so the idea is that you can have a bunch of mutations you want to do, and in a similar fashion here, it then creates the different constructs. And here you can then add your base construct, so to speak, with the termini, truncations, and which fusion site. And what's then powerful is then you can then get all these sequences exported, for example, to Excel, and then and start working on getting those expressions. So that's the construct and experiment design menus here. And there's then also these sites which summarize different truncation sites, fusion sites, and loop deletions, as also can be seen in the figures from the actual manuscript.